Yeah. Okay, so welcome back. सबने चाय पी ली? नींद खुल गई? All right, okay. Please show the last slide. Last slide तुम्हारे सामने ही तो है. हाँ वसंत, only the counter current is left. So let's go on to बड़े बच्चे छोटे बच्चों को तंग नहीं करें. Name of the topic after lung kidney isthma. Okay. Okay, all right. Now this is as far as your let's go on to the last topic in kidney, and that is the counter current mechanism. How is new bicarbonate found in the collecting duct? Jennifer, this is not related to the filtered bicarbonate. That's why we called it a new bicarbonate. Okay? All right. So let's go on to the last topic in kidney, and that is the counter current mechanism. This is going to be the counter current mechanism. Now, as far as counter current mechanism is concerned, please, there are very few questions which come from counter current. Counter current, very straightforward questions because nobody can ask you to explain. So counter current is mainly for explanation purposes. Few questions from counter current, very straightforward questions which you will be able to answer. So this is more for understanding, right? Let's see now the now counter current. What do you understand by counter current? Counter current is whenever you have two vessels which are close to each other, parallel to each other, and with flow in opposite directions. That is called a counter current, right? Counter current. This is your two vessels close to each other. Two vessels close to each other, parallel to each other, two vessels close to each other, parallel to each other, with flow in opposite directions, right? With flow in opposite directions, that is known as a counter current mechanism, right? With flow in opposite directions is known as, these are two vessels which are close to each other, parallel to each other, with flow in opposite directions. Where do I see a counter current mechanism? Counter current mechanism is seen in the kidney. This is seen in the kidney. We will be doing that in detail. You can see the counter current mechanism in the kidney. You can also see a counter current mechanism in the testicular vessels. This is seen in the testicular vessels. Counter current in the testicular vessels. You also see a counter current in the intestinal villi. Intestinal villi. You see a counter current mechanism in the intestinal villi. Each intestinal villus will show you a counter current mechanism. You also see counter current in what is known as the veni comitantes. Veni comitantes. And what is this veni comitantes? Veni comitantes are a pair of veins. Pair of veins which are associated with certain arteries. Pair of veins which are associated with certain arteries. That is, and what are they involved in? They are involved in conserving heat. Yes. So these are the areas where you see the counter current mechanism. You see it in the kidney, you see the testicular vessels, in the intestinal villi, in the veni comitantes. What is veni comitantes? Veni comitantes are a pair of veins associated with certain arteries, and they are involved in conserving heat, temperature regulation. Yes. Sai Spandana Reddy, brass mechanism, we will complete it in cardiovascular. Okay? All right, so this is your counter current mechanism. Now let us see which are the counter current in the kidney. What is the counter current in the kidney? Counter current in the kidney, there are two. There is a counter current. There is a counter current multiplier. There is a counter current multiplier and a counter current exchanger. Counter current multiplier and a counter current exchanger. What is the countercurrent multiplier? Countercurrent multiplier is the loop of Henle. What is the countercurrent exchanger? This is the Vasa recta. Loop of Henle, this creates the medullary interstitial hyperosmolarity. It creates the medullary interstitial hyperosmolality. Right? It creates the medullary interstitial hyperosmolality. There are two countercurrents in the kidney. There is a countercurrent multiplier and a countercurrent exchanger. Countercurrent multiplier is a loop of Henle which creates medullary interstitial hyperosmolality, and countercurrent exchanger, which is the Vasa recta. And Vasa recta will maintains the medullary interstitial hyperosmolality. One creates it and the other maintains it. Right? One creates it and the other maintains it. Right? This is the countercurrent multiplier and the countercurrent exchanger. Abhishek, aorta, IVC, both are close to each flow in opposite. But countercurrent kiske liye? What will they do? A countercurrent has to achieve something, na? Yes? In the testicular vessels, it helps to maintain scrotal temperature lower than body temperature. Veni comitantes help to conserve heat. Intestinal villi, the countercurrent will help in absorption of nutrients. In the kidney, there is a countercurrent multiplier and a countercurrent exchanger. Have you understood? Right? Loop of Henle is LOH is loop of Henle. This creates the medullary interstitial hyperosmolality, and Vasa recta will maintain the medullary interstitial hyperosmolality. Right? So this is your countercurrent in the kidney. Two countercurrent mechanisms. Now let's see what is the countercurrent multiplier. How does it create the medullary interstitial hyperosmolality? Now this is very easy to understand. The two limbs of loop of Henle. The two limbs of loop of Henle, they have a differential permeability. Right? They have a differential permeability. The two limbs of loop of Henle have a differential permeability. How differential permeability? The descending limb of loop of Henle, is permeable to only water, and the thick ascending limb is permeable to only solutes, Na+, K+, and Cl-. So this is what is meant by differential permeability. The descending limb is permeable only to water, ascending limb only to solutes, Na+, K+, to Cl- co-transport. And if you remember, we said the absorption of solutes in the thick ascending limb is more than the absorption of water in the descending limb, right? So what happens is inside the fluid will become hypotonic, but outside the interstitial, this is the interstitial fluid which surrounds this loop of Henle. This interstitial fluid will now become concentrated. Why will it become concentrated? Because there is now less movement of water, more movement of solutes into the interstitium, from the lumen into the interstitium. So the fluid inside the lumen will become dilute, but the interstitial fluid will become hypertonic. So this is helping you, this differential permeability is helping to create a hyperosmolar medullary interstitium, right? It creates a hyperosmolar medullary interstitium, right? So this is because of the differential permeability of the two limbs of loop of Henle. The descending limb will absorb water and the ascending limb will absorb solutes, but the absorption of solutes is much more than the absorption of water. So the surrounding fluid which surrounds the loop of Henle, that is the interstitial fluid, that becomes hyperosmolar medullary interstitium. This is created by the loop of Henle. Now, one important point, why do I need a hyperosmolar medullary interstitium? Now, if you look at this, if you look at the kidney, there is the cortex, right? And there is the medulla. The cortical interstitium has an osmolality of 300 milliosmoles per liter, which is isotonic. 300 milliosmoles per liter, which is isotonic, right? As you keep going deep into the medulla, the osmolality increases, and deep in the medulla, so as you keep going deep into the medulla, the osmolality will increase. It is 600, 900, 1200. Deep in the medulla, it is 1200 milliosmoles per liter. Remember, plasma has an osmolality of 290. I'm taking it as 300. But deep in the medulla, the osmolality of the interstitium is now 1200. Why is that so? Mainly because of the differential permeability of the loop of Henle. There is movement of water from the lumen into the interstitium in the descending limb, and there is movement of solutes from the ascending limb into the interstitium, but movement of solutes is more. So interstitial fluid becomes concentrated. This is the medullary interstitial osmolality. Now the question arises, why do I need this? What is the significance of a hyperosmolar? Significance of a hyperosmolar medullary interstitium. What is the significance of a hyperosmolar medullary interstitium? Hyperosmolar medullary interstitium, it helps to form a concentrated urine. This is involved in concentration of urine. How? Now, if you look at the collecting duct, there is a cortical collecting duct, there is an outer medullary collecting duct, inner medullary collecting duct, which opens onto a renal papilla. From renal papilla, the urine will flow into the minor calyx, major calyx, renal pelvis, ureter, urinary bladder, urethra, and out. So the tubular fluid drains into the collecting duct. So collecting duct, you've got cortical, you've got outer medullary, you've got inner medullary collecting duct, right? Now, to produce concentration of urine, two things are required. For concentration of urine, number one, there has to be the presence of ADH. ADH will increase the aquaporin 2 on the luminal side of, it increases the aquaporin 2 on the collecting duct cells, right? P cells of the collecting duct, yes. It increases the aquaporin 2 on the P cells of the collecting duct, which increases the water reabsorption, right? And number two, you will also need a medullary interstitial hyperosmolality, because please remember, you will need a medullary interstitial hyperosmolality. See, because you must remember that water always moves from a dilute to a concentrated solution. So if the fluid which surrounds the collecting duct is concentrated, only then the water will move from the lumen into the interstitium. Have you got my point? I need the interstitial fluid which surrounds the collecting duct should be concentrated, so that there is, see, out of these two, if one is absent, if there is no ADH, no concentration of urine. If medullary interstitium hyperosmolality is not present, no concentration of urine. For concentration of urine, I need both the things. I need ADH and I need a medullary interstitial hyperosmolality. Have you understood? See, somebody, Vani Dhawan, you've written that blood vessels. Now, of course, there are blood vessels present, but between the collecting duct and the blood vessels, there is interstitial. Interstitial fluid will be present. Have you understood? Right? There will be presence of an interstitial fluid. Now, this interstitial fluid should be concentrated. Only then the water will move from the lumen of the collecting duct into the interstitium and then into the blood. Have you understood? So this is for concentration of urine, I need both the things. So that is why I need a medullary interstitial hyperosmolality. So that means, what can be the maximum urinary osmolality? What is the maximum urinary osmolality that can be achieved? Maximum urinary osmolality will be 1200 milliosmoles per liter. This is the maximum urinary osmolality which can be achieved. Urine can never be more concentrated than 1200. इससे ज़्यादा तो हो ही नहीं सकता, because that is the maximum osmolality of the fluid surrounding the collecting duct. So this is going to be, it cannot be more than 1200. Some books have said this is 1400. Not 1200, 1400. But generally acceptable figure is 1200. Okay? So this can be 1400 as well. Maximum urinary osmolality. Now, the next important question is, what is going to be the range of urinary osmolality? What is the range of urinary osmolality? Now, there are two questions over here. What is the usual range of urinary osmolality, and what is the possible range of urinary osmolality? The usual range of urinary osmolality is 300 to 1200 milliosmoles per liter. That means it is isotonic to hypertonic. What is the possible? Usually the urine which is produced is isotonic to hypertonic. Cortical nephrons will produce an isotonic urine. Yes. Usually it is isotonic to hypertonic. But what is the possible range? Possible range of urinary osmolality, this can be 50 to 1200 milliosmoles per liter. When do you get a urinary osmolality of 50? When there is no ADH. ADH is absent. When do you get a urinary osmolality of 1200? When you have a maximum ADH. So possible range is 50 to 1200, but the usual range, urine is usually isotonic to hypertonic, 300 to 1200 milliosmoles per liter. Possible 50 to 1200 milliosmoles per liter. Right? So this is the role of the loop of Henle. What does the loop of Henle do? Loop of Henle helps to produce a hyperosmolar medullary interstitium. How does it do so? This is because the two limbs of loop of Henle have a differential permeability. There is absorption of water from the descending limb, absorption of solutes from the ascending limb. But the absorption of solutes is much more than the absorption of water. So the fluid which surrounds a loop of Henle becomes concentrated or hyperosmolar. What is the significance of a medullary interstitial hyperosmolality? Helps to produce a concentration of urine. For concentration of urine, two things are required. I need the presence of ADH. ADH will increase the number of aquaporin 2. It increases the number of water channels in the collecting duct, but water will only move if the fluid surrounding these water channels, fluid surrounding the collecting duct is concentrated. So for concentration of urine, you need ADH and you need a medullary interstitial hyperosmolality. ADH will increase the number of aquaporin 2 on the luminal side, and it will, and number two, we also need a medullary interstitium. Water will only move from a dilute to a concentrated area. What is the maximum urinary osmolality? 1200. Some books have said this is 1400. What is the range of urinary osmolality? 300 to 1200, but a possible range is 50 to 1200. 50 is when there is no ADH, 1200 when there is maximum ADH. So this is a role of the loop of Henle, right? Now, loop of Henle, like I said, is involved in creating the medullary interstitial hyperosmolality, but Vasa recta is involved in maintaining it. So my next important question related to loop of Henle is, this is, what are the solutes responsible? Which are the solutes responsible for the medullary interstitial hyperosmolality? Which are the solutes which are responsible for medullary interstitial hyperosmolality? This is going to be sodium, potassium chloride from the thick ascending limb, and also urea. Urea will be from the medullary collecting duct. Medullary collecting duct. Advanced collecting duct. Okay? See, please try and understand. This is, if this is, let's say, a cross section of the collecting duct. This is the collecting duct cells. Right? Let me just try and make a bigger diagram here. This is the collecting duct cells here. Yes. Aquaporin 2 is on the luminal side. This is aquaporin 2. Aquaporin 3 and 4. Aquaporin 3 and 4 are on the basal side. Here you have the interstitial fluid surrounding the collecting duct. If inside fluid is at inside the collecting duct, yes, if this is concentrated, if this is the interstitial, medullary interstitial, this is 1200, let's say 1200 milliosmoles concentrated. If this is 1200, then water will move from, yes, this is aquaporin 2, aquaporin 3 and 4 on the basolateral membrane. So water will move from the lumen into the cells and then into the interstitium. So water will always move from a dilute to a concentrate till the osmolality is the same on both sides. That is why I need the medullary interstitium to be concentrated. How is that concentration achieved? By 4 solutes. Sodium, potassium chloride, which will come from the thick ascending limb into the interstitium, and urea, which will come from the medullary collecting duct. Medullary collecting duct. Please remember, ADH will increase the number of aquaporin 2 on the luminal membrane. Aquaporin 3 and 4, their number is fixed. These are constitutionally present. Aquaporin 3 and 4 are constitutionally present. What is constitutionally present? That means the number is fixed. ADH will increase the number of aquaporin 2 on the luminal side. A hypertonic medullary interstitium. So both the things will be required. ADH will increase the number of aquaporin 2, and medullary interstitial hyperosmolality is the reason why water will move from the lumen into the interstitium. Have you understood? This is your countercurrent mechanism. Okay? Okay. Now, the next important point, what are the solutes responsible for medullary interstitial hyperosmolality? Sodium, potassium chloride, this is from the thick ascending limb, and urea, which is from the medullary collecting duct. Right? This is from the medullary collecting duct. कसम शेट्टी चंदू, पीसीटी कहाँ है? कॉर्टेक्स में या मेडुला में? पीसीटी से जो यूरिया अब्सॉर्ब होगा will go into the peritubular capillaries. PCT is in the cortex. Have you understood? We are interested in the medulla, isn't it? This is what is MCD. Like I said, this is the medullary collecting duct. Okay. Now let us see what is the urea handling by the kidney. Let's see how does the urea handling by the kidney take place. Yes, urea handling by the kidney. Now what happens in the urea handling by the kidney? Let's see this. Now, urea is a freely filtered substance, right? This is a freely filtered substance. Urea is a freely filtered substance. And when I say it is a freely filtered substance, yes, it enters into the glomerular filtrate. This is urea, freely filtered substance. Now, 52% of the urea will be reabsorbed in the PCT, right? 52% of the urea will be reabsorbed in the PCT. Okay? It is reabsorbed in the PCT. It goes back into the blood. Please remember, this is not what is responsible for medullary interstitial osmolality. After this, there is no reabsorption of urea anywhere. No reabsorption of urea anywhere, but there is going to be reabsorption of water. So by the time it reaches into the medullary collecting duct, what will happen to the urea concentration? There is an increase in the urea concentration. Why is there an increase in the urea concentration in the medullary collecting duct? Because what has happened is there is no reabsorption of urea anywhere, but there has been reabsorption of water. So urea concentration in the tubular fluid now increases. Medullary collecting duct has the presence of urea transporters. There is UTA1, UTA3, these are urea. If you remember, we have discussed in the first chapter that the transport of urea is by passive transport, by facilitated diffusion. There are urea transporters which are present in the collecting duct. So what happens is the urea will move from the collecting duct into the interstitium, because now urea in the medullary collecting duct is in a higher concentration and it now comes into the interstitium. So when it comes into the interstitium, it will now increase interstitial osmolality. It will increase the medullary interstitial osmolality. Yes, this will come from the collecting duct into the interstitium. How does it do so? Because medullary collecting duct is permeable to urea. It has the presence of urea transporters, which are the subtypes of urea, UTA1 and UTA3. It increases, like I said, it increases the medullary interstitial osmolality. It increases the medullary interstitial osmolality, contributes to make the medullary interstitium concentrated. Now, the descending thin segment also has the presence of urea transporters. So when urea concentration increases in the interstitium, it will go back into the nephron via these urea transporters on the thin segment. It goes back to the tubular fluid. Yes, the remaining part of the nephron is impermeable to urea. The thick ascending limb, the DCT, the cortical collecting duct, this is impermeable to urea. So there is no movement of urea here. Then it reaches into the medullary collecting duct. Some amount of the urea goes into the interstitium and about 20% of the filtered urea is excreted out. Some of it goes into the interstitium, some is excreted out. Increases the medullary interstitial hyperosmolality. When the concentration of urea increases in the interstitium, it goes back into the tubule through the urea transporters on the descending thin segment. Now by the time more urea comes from the PCT, it goes in the thick ascending limb, DCT, cortical collecting duct, reaches medullary collecting duct. Some urea comes into the interstitium, some is excreted out. Increases the medullary interstitial hyperosmolality, goes back into the descending thin segment, thick ascending limb, DCT. This entire process is known as urea cycling. Urea cycling. Urea is being cycled here. Advantage of urea cycling. What is the advantage of urea cycling? Advantage of urea cycling, it increases the medullary interstitial hyperosmolality. It increases the medullary interstitial hyperosmolality. And do I need any energy for urea absorption? No. No energy is used. Remember, sodium potassium chloride, no energy is used here because the movement of urea is by passive transport. Please understand, I can increase the medullary interstitial osmolality by increasing the absorption of sodium potassium chloride, but that is active. It needs energy. Urea will increase the medullary interstitial osmolality without consuming energy. Have you understood? This is urea cycling. But if you are asked a question, which are the most important solutes? Most important for medullary interstitial hyperosmolality is sodium potassium chloride. Most important is just 1 second, please. Let me log into the app. I've also been logged out. Okay, just 1 second. Okay, there we are. Okay, all right. So this is the, like I said, where was that? Most important solutes responsible for medullary interstitial hyperosmolality. Most important solutes responsible for the osmolality is going to be sodium potassium chloride. Yes, these are the important solutes. But yes, what contributes, what increases the medullary interstitial hyperosmolality is urea. Yes, urea also increases. Now, what is urea cycling? Let's see this once again. What happens in urea cycling? By the time the fluid reaches the medullary collecting duct, it is conjugated. There is high urea concentration. Urea moves from collecting duct into the interstitium via urea transporters, examples of facilitated diffusion. This urea will increase the medullary interstitial osmolality. When urea concentration increases in the interstitium, it goes back into the nephron through the descending thin segment. Descending thin segment also has urea transporters. By the time more urea comes from the PCT, the urea now moves from descending thin segment to thick ascending limb, impermeable to urea, DCT, impermeable to urea, cortical collecting duct, impermeable to urea, reaches the medullary collecting duct. Medullary collecting duct is permeable to urea, right? 20% of filtered urea is excreted, remaining goes back into the interstitium. This is known as urea cycling. Yes, important points. Which parts of the nephron are permeable to urea? Which parts of the nephron are permeable to urea? Which parts of the nephron are permeable to urea? PCT, descending thin segment, and the medullary collecting duct. Which are impermeable to urea? Which are impermeable to urea? Which are impermeable to urea? Thick ascending limb, DCT, and cortical collecting duct, right? Cortical collecting duct. right? So permeable to urea, PCT, descending thin segment, medullary collecting duct. Impermeable to urea will be thick ascending limb, DCT, and the cortical collecting duct. Now somebody said, if the descending thin segment is permeable to urea, then why does urea not go from descending thin segment into interstitial? Please remember, urea will only move if there is. Urea will only move if there is um, only moves from higher to lower concentration. It cannot go from lower to higher concentration, even if urea transporters are present. Urea transporters are examples of passive transport. Passive transport, right? So this is as far as your which are the ones which are permeable to urea is concerned, right? Impermeable to urea, thick ascending limb, DCT, and cortical collecting duct. If you're asked a question, where does urea reabsorption happen? Urea reabsorption Urea reabsorption is in the PCT, not in the medullary collecting duct. What happens in the medullary collecting duct is not a reabsorption. It is a urea cycling. An advantage of urea cycling is to increase the medullary interstitial hyperosmolality, right? Ascending thin segment. Ascending thin segment as well. Descending thin segment and the ascending thin segment. Yes. And advantage of urea cycle. Urea reabsorption is only in the PCT, but the urea cycling is going to be advantage of urea cycling to increase the medullary interstitial hyperosmolality. We've done that already. Okay, let me see. Okay, I can't follow your questions. There's too much of writing. Ascending thin segment is also permeable to urea. Ascending thin segment is also permeable to urea. Okay. Urea transport is by urea transporters. There are different types of urea transporters which are present in the nephron. Not important. What was important is UTA1 and A3 in the medullary collecting duct. Otherwise, you have UTA1, A2, A3, A4. There's nothing else specific about the urea transporters, but in the collecting duct, yes, it is UTA1 and A3. Okay? Okay. Most important solute responsible for medullary interstitial osmolality is sodium potassium chloride. Urea from medullary collecting duct will increase the medullary interstitial osmolality, but is not the most important solute. Okay. All right. So this is as far as your urea handling by the kidney is concerned. Next, let's have Let's have a look at the role of the vasa recta. What is the role of the vasa recta? Prutu Chandan, it is via urea transporters, facilitated diffusion. Facilitated diffusion. When I say permeable to urea means they have the presence of urea transporters. That is how they are permeable. Permeable to urea means urea transporters are present. Urea carrier proteins for movement of urea. Carrier proteins for the movement of urea. Thick ascending limb, ascending thin segment. CP, क्या बोल रहे हो बेटा? Okay. Okay. Let's have a look at the role of the vasa recta. Vasa recta involved in maintaining the medullary interstitial hyperosmolality. So let's have a look at the vasa recta. Now, some important points about the vasa recta, and then we can go on to its role in maintaining. Now, vasa recta, these are countercurrent with each other and with the They are countercurrent with each other and with the loop of Henle. The two limbs of Vasa recta, are countercurrent with each other. The two limbs of Vasa recta are countercurrent with each other and with the loop of Henle. Right? So this is the first important point. Second important point, both limbs of Vasa recta, are freely permeable. Both limbs of Vasa recta are freely permeable to solutes and water. Both limbs of Vasa recta are freely permeable to solutes and water. This is very, very important because if you remember, cell membranes are actually semi-permeable, but Vasa recta is very unique because both limbs are freely permeable to solutes and water. Next important point, Vasa recta receives less than 5% of the renal blood flow. 95% of the renal blood flow goes into the cortex. Vasa recta receives less than 5% of renal blood flow, so it has a very, very, very slow rate of blood flow. The blood flow rate through the Vasa recta is very, very slow, and this helps in the exchange mechanism. Right? Both limbs of Vasa recta are freely permeable to solutes and water. They receive less than 5% of the renal blood flow, and they have a very, very slow rate of blood flow, which helps in the exchange mechanism. Then another important point, Vasa recta are the only supply of blood. These are the only supply of blood and therefore oxygen to the medulla. And this was an AIMS question which was asked in 2017, the supply of blood to the medulla, supply of blood and therefore oxygen to the medulla is by the Vasa recta. This is the only supply. So these are four important points about Vasa recta which you have to keep in mind, and then we discuss what is the role of the Vasa recta. The two limbs of Vasa recta, these are countercurrent with each other and with the loop of Henle. Number two, both limbs of Vasa recta are freely permeable to solutes in water. Vasa recta has a very, very, very slow rate of blood flow, which helps in the exchange mechanism. And Vasa recta is the only supply of blood and oxygen to the medulla. So let's look at the first thing here. We said Vasa recta is countercurrent to each other and countercurrent to the loop of. Discuss this. This is the nephron here. Right? The nephron here. And let's see now what happens here. If you look at this is the afferent arteriole. Yes, afferent arteriole. It forms the glomerular capillaries, which forms the efferent arteriole. Afferent arteriole will form the glomerular capillaries, which forms the efferent arteriole. Then they form the peritubular capillaries. These are capillaries around the PCT, peritubular capillaries. Yes. Now, the peritubular capillaries will form the descending limb of the Vasa recta. Now, the descending limb of the Vasa recta is associated with the ascending limb of loop of Henle. Descending limb of the Vasa recta is associated with the ascending limb of loop of Henle. Then, this Vasa recta also makes a U-shaped turn. Now, the ascending limb of Vasa recta, this is associated with the descending limb of loop of Henle. I made you write, I said the two limbs of Vasa recta, they are countercurrent with each other and with the loop of Henle. Right? They are countercurrent with each other and with the loop of Henle. The two limbs of Vasa recta are countercurrent with each other and with the loop. So you can see that they are not just, they are countercurrent. So descending limb of the Vasa recta is associated with the ascending limb of loop of Henle, and ascending limb of Vasa recta is associated with the descending limb of loop of Henle. And then this Vasa recta will form the capillaries around the DCT and around the collecting duct. Yes, and then of course it forms a renal vein and venules and so on, and forms a renal vein. Right? So this is as far as your very, very unique arrangement of the Vasa recta is concerned. Yes, the two limbs of Vasa recta, these are countercurrent with each other and with the loop of Henle. Very, very slow rate of Vasa recta blood flow. Number 3, I told you this is the only supply of oxygen and therefore blood and therefore oxygen to the medulla. Right? This is as far as your important points about Vasa recta. Now, what is this? What is the significance of this countercurrent mechanism that you see here? Now, what happens is, remember, solutes which are reabsorbed from the thick ascending limb will enter into the descending limb of the Vasa recta. Yes, they enter into the descending limb of the Vasa recta. And what is the direction of movement of these solutes? Towards the medulla. Water which is absorbed from the descending thin segment, enters into the ascending limb of Vasa recta. And what is the direction of movement of blood in the ascending limb of Vasa recta? Away from the medulla into the cortex. Right? So keep this in mind. Solutes are moving towards the medulla, and water is moving away from the medulla towards the cortex. So this is a very simplified version of what is the role of Vasa recta. Let me do the role of Vasa recta in maintaining the medullary interstitial hyperosmolality in a little more detail. Let's see this. When I look at the Vasa recta, yes, the blood which enters into the Vasa recta comes from the cortex. What is the interstitial osmolality? Cortical interstitial osmolality is 300. As you keep going into the medulla, the interstitial osmolality increases, becomes 600. What is contributing to this increase in interstitial osmolality? Sodium potassium chloride and urea from the medullary collecting duct. And deep in the medulla, this is going to be 1200 milliosmoles per liter. Right? Now, the blood which comes into the descending limb of Vasa recta, has an osmolality of 300 because this is coming from the cortex. Right? It is at 300. It is coming from the cortex. Yes? Now, as it moves into the descending limb, the surrounding osmolality is now increasing. And remember, the limbs of Vasa recta are freely permeable to solutes and water. So what happens is, solutes will now enter into the Vasa recta from the interstitium into the Vasa recta. Solutes are higher outside, less inside, so they come from outside to inside. Water is more inside, less outside, so water tends to flow from inside to outside. This is solutes here. And this is water. Yes? This will happen till the inside and the outside osmolality is the same. Yes? So it becomes 600 now. Now this moves into an area where surrounding is even higher. Surrounding osmolality is even higher. So more solutes will come from outside to inside. Water goes from inside to out till the inside and the outside osmolality is the same. Yes? Again, this moves into an area where surrounding osmolality is even higher. So more solutes will come from outside to inside. Water goes from inside to out till the inside and the outside osmolality is the same. This now the blood, as it moves into the ascending limb, is going from higher. Now it is inside the blood is at a higher osmolality, outside is lower. So solutes go back into the interstitium and water enters into the blood. This again goes into an area where surrounding interstitial osmolality is lower. More solutes go back into the interstitium and water will enter into the blood. This is what is known as your counter current exchanger. What is the advantage of this counter current exchanger? Solutes will enter into the vasa recta from the surrounding interstitium in the descending limb. But in the ascending limb, these solutes are dumped back into the interstitium. So this prevents a washout of the solutes. Have you understood? It prevents a washout of the solutes. In the descending limb, solutes are entering into the vasa recta. In the ascending limb, they are going back into the medulla. This prevents a washout of the solutes. Yes. In fact, there is slight loss of solutes, obviously. It is not 100% accurate. So the osmolality of the blood which leaves the vasa recta is at about 325. So these solutes are being replenished. There is loss of some solutes, but these solutes are being replenished by the loop of Henle and by the urea. Have you understood? So this is a dynamic process. So you must keep in mind the loop of Henle is creating this interstitial osmolality, hyperosmolality. Urea is contributing to it. Vasa recta is maintaining it. Vasa recta, see, what is this U-shaped arrangement of the vasa recta? Why is it important? Because remember, vasa recta is the only supply of oxygen to the medulla. Medulla needs oxygen. So vasa recta has ensured oxygen supply to the medulla, but at the same time, because of this U-shaped arrangement, vasa recta has ensured that majority of the solutes remain within the medulla. This is the role of the vasa recta. The questions which come from countercurrent mechanism are very, very simple. What is the countercurrent multiplier? Loop of Henle. What is the countercurrent exchanger? Vasa recta. Solutes responsible for medullary interstitial hyperosmolality: sodium, potassium chloride, and urea. Where does urea come from into the interstitium? From the medullary collecting duct. Are we clear with that? Right? This is as far as your very, very important. These are the which come from the countercurrent mechanism. Right? Loop of Henle creates it. Vasa recta will maintain it. Loop of Henle, which parts of the loop of Henle are in the countercurrent? Descending thin segment and thick ascending limb. Ascending thin segment is not a part of the countercurrent mechanism, right? Loop of Henle, please remember, ascending thin segment is not a part of the countercurrent. Countercurrent is descending thin segment and thick ascending limb, not the ascending thin segment, right? It is not a part of the countercurrent. Yes, it is involved in urea cycling, but it is not. Urea cycling is not countercurrent. Countercurrent is the differential permeability of the two limbs of loop of Henle. Descending thin segment for water, thick ascending limb for solutes, more absorption of solutes from the thick ascending limb. Okay? All right. ठीक है. See, vasa recta का रोल इंपॉर्टेंट इसलिए है क्योंकि vasa recta ने ऑक्सीजन सप्लाई करना है, साथ साथ इंटरस्टिशियल की ऑस्मोलैरिटी को मेंटेन करना है. How does it maintain? Is with the U-shaped arrangement. अगर ये straight vessel होता तो ये सारे के सारे solutes venous side पे चले जाते. Have you got my point? Right? So this is going to be the countercurrent mechanism. Okay? Questions are very simple from countercurrent: multiplier, loop of Henle, vasa recta, exchanger. Okay? Loop of Henle का कौन से points? ATS is DTS and thick ascending limb. Yes, absolutely right. Chalo. Let's go on to our next chapter. Everybody's okay? Alive? Okay. ATS cross इसलिए है क्योंकि it is not a part. ATS is not a part of not part of the counter current multiplier. It is not a part of the counter current multiplier. How compensated, Abhishek? क्योंकि continuous dynamic process है ना. Sodium potassium chloride तो reabsorb हो ही रहे हैं thick ascending limb से. Have you understood? ठीक है? Okay. All right. Chalo, let's go on to our next system. Let's see the next system that we're going to do over here, and that is going to be the respiratory system. Let's see the respiratory system. Limiting pH 4.5. Racy, no क्या है? Okay, all right. So this is your let's see the respiratory system here. Okay, let's start with the next system. Respiratory, we are already 10 minutes late. Chalo, okay. Okay, then okay. Bus, stop typing now. Bus, enough. All right. So let's see the first thing as far as the respiratory system is concerned, and that is, what are the agents which are going to cause a bronchodilation? Which are the ones which cause a bronchodilation, and which are the ones which cause a bronchoconstriction? अरे बेटा, RAS CVS में हो जाएगा. RAS will be done in CVS. ठीक है? Bronchodilation and bronchoconstriction. What are the agents which will cause a bronchodilation and a bronchoconstriction? Now, as far as bronchoconstriction and bronchodilation, sympathetic discharge is responsible for adrenaline. Adrenergic group of drugs are responsible for a adrenergic group of drugs adrenergic agents will be responsible for a bronchodilation. What is bronchoconstriction? Bronchoconstriction is parasympathetic acetylcholine cholinergic agents. Right? Cholinergic agents, bronchodilation and bronchoconstriction. Now, another one which causes a bronchoconstriction is substance P. Substance P is an inflammatory chemical. It causes a bronchoconstriction. Substance P. Then adenosine. Adenosine will also produce a bronchoconstriction. Adenosine. It produces a bronchoconstriction. Cool air also produces a bronchoconstriction. Adenosine, substance P, these are inflammatory chemicals. They will produce a bronchoconstriction. Cool air will also produce a bronchoconstriction. In winters, there is increased incidence of asthma. Right? There is increased incidence of asthma, which is seen in winters, because cool air itself is a bronchoconstrictor. So substance P, adenosine, this produces, cool air itself is a bronchoconstrictor. On the other hand, VIP, vasoactive intestinal peptide, this is secreted by NANC nerves. NANC nerves. What is NANC nerves? This is non-adrenergic. Non-adrenergic, non-cholinergic nerves. Non-adrenergic, non-cholinergic nerves. Right? VIP, which is secreted by NANC nerves, this is called non-adrenergic, non-cholinergic nerves. VIP is a potent bronchial smooth muscle. Bronchial smooth muscle, relaxant. This is a potent bronchial smooth muscle relaxant. Right? So this is your, what are the agents which cause a bronchodilation and bronchoconstriction? Sympathetic, adrenaline, adrenergic agents VIP. These are VIP, which is secreted by NANC. NANC is non-adrenergic, non-cholinergic nerves, right? And this is a potent smooth muscle relaxant, bronchial smooth muscle relaxant. Bronchoconstriction produced by parasympathetic stimulation. Parasympathetic causes a bronchoconstriction. Acetylcholine, bronchoconstriction. Cholinergic agents, bronchoconstriction. Substance P, adenosine. Cool air itself is a bronchoconstrictor, but VIP is a bronchodilator. Right? So these are the agents which cause bronchodilation and bronchoconstriction. The next important point, to some important formulae. What do you understand by total or pulmonary ventilation? Total or pulmonary ventilation. Total or pulmonary ventilation. This is respiratory rate into the tidal volume. This is also sometimes called the minute ventilation. Minute ventilation. Right? Total or pulmonary ventilation, this is respiratory rate into the tidal volume. Yes. What is the alveolar ventilation? What is alveolar ventilation? Alveolar ventilation is respiratory rate into tidal volume minus the dead space, right? Respiratory rate into tidal volume minus the dead space. That is known as alveolar ventilation, right? Respiratory rate into tidal volume minus the dead space. Right? Total or pulmonary ventilation, respiratory rate into tidal volume. What is alveolar ventilation? Alveolar ventilation is respiratory rate into tidal volume minus the dead space. Yes. Next, let's have a look at, by the way, this has been asked as a question. What is minute ventilation? Respiratory rate into the tidal volume. Let's have a look at the dead space. What is dead space? Now, dead space is volume of air which does not participate, volume of air which does not participate in respiratory exchange. This is known as dead space. Volume of air which does not participate in respiratory exchange. This is known as the dead space. And how much is normal dead space? Normal dead space is about 140 to 150 ml in an adult, which is about 2 ml per kg body weight, or 2 ml per kg. This is what you see in an adult. This is the dead space. This is the volume of air which is present in the trachea, in the bronchi, which is not participating with respiratory exchange. That is known as the dead space. I've done two important formulae up till now, and that is what is total pulmonary ventilation, respiratory rate into the tidal volume. What is alveolar ventilation? Alveolar ventilation is respiratory rate into tidal volume minus the dead space. Now, my next important point was, what is dead space? Dead space is the volume of air which does not participate in respiratory exchange. And how much is the normal dead space volume? This is 140 to 150 ml in the case of an adult, or 2 ml per kg. Now, there are two types of dead space. There is a physiologic dead space and there is a anatomic dead space. So let us see what is the relationship between the two. The physiologic or the total dead space. Physiologic or the total dead space, this is equal to the anatomic dead space. Anatomic dead space plus a wasted ventilation. Right? This is an anatomic dead space plus a wasted ventilation. What is physiologic or physiologic and the anatomic dead space. Anatomic dead space, physiologic dead space is anatomic plus a wasted ventilation. Now, let's focus on what is wasted ventilation, right? What is wasted ventilation? Now, wasted ventilation is wasted ventilation is seen whenever the VQ ratio is more than 1. That is called a wasted ventilation. So you must understand, at a VQ ratio of 1, that means ventilation and perfusion, when they are equally matched, when they're equal to each other, at a VQ ratio of 1, right? VQ ratio of 1 is taken as ideal. This is an ideal VQ ratio. Why ideal? Why ideal? Because you have maximum oxygenation of the blood is achieved at a VQ ratio of 1. Hemoglobin is 100% saturated at a VQ ratio of 1, right? Maximum oxygenation is achieved at a VQ ratio of 1. Hemoglobin is 100% saturated. VQ ratio of more than 1 means what? This means ventilation is in excess of. Ventilation is in excess of the perfusion. That means now the ventilation is more than the perfusion, is in excess of the perfusion. That means this extra ventilation that we are doing, that is being wasted. Why wasted? Because hemoglobin saturation is already 100% at a VQ ratio of 1. At VQ ratio more than 1, when ventilation is in excess of the perfusion, VQ ratio more than 1 means that this extra ventilation is being wasted, because hemoglobin saturation cannot increase from 100% to 150 or 170%. It remains at 100. Cannot become more than 100. 100 is achieved at 1. VQ ratio more than 1 means wasted. It is being wasted. It is not helping me to increase the hemoglobin saturation. So wasted ventilation, more than 1. And how can you get wasted VQ ratio more than 1? If you have, number 1, over-ventilated alveoli. If you have over-ventilated alveoli, that means ventilation has increased without increasing the perfusion. VQ ratio is more than 1. Over-ventilated alveoli, or if there are under-perfused alveoli. Over-ventilated alveoli or under-perfused alveoli. That means ventilation is normal, but perfusion is less than normal. Let's say ventilation is normal, but perfusion has become half. Something has happened. 1 divided by half is 2, more than 1. Wasted ventilation, right? So this is what is, whenever you have a wasted ventilation, whenever you have a VQ ratio more than 1, we call it a wasted ventilation. And whenever there is a wasted ventilation, that means there is going to be an increase in the physiologic dead space. Have you understood, right? Physiologic dead space is anatomic plus wasted ventilation. Wasted ventilation, when do you see wasted ventilation? Whenever the VQ ratio is more than 1. Why VQ ratio more than 1 is called wasted ventilation? Because at VQ ratio of 1, you have achieved maximum oxygenation, maximum hemoglobin saturation of 100. VQ ratio more than 1 means ventilation is in excess of the perfusion. This extra ventilation is not going to increase the hemoglobin saturation to more than 100%. Not possible. So this extra ventilation is being wasted. How do you get a wasted ventilation? How do you get a VQ ratio more than 1? Over-ventilated alveoli or under-perfused alveoli. If perfusion is compromised, right, then there is a wasted ventilation. Yes. For instance, now please remember, like for instance, there was a question which was asked, and that is that here is an area of the lung. There is an area of the lung which has a VQ ratio of 10. VQ ratio of 10. Something has happened. VQ ratio of 10. Maybe there is a thrombus in the pulmonary blood, in the blood vessel, perfusion has reduced, and VQ ratio is now 10 in this segment. So there is an increase, it causes an increase in anatomic dead space or the physiologic dead space. Anatomic dead space or physiologic dead space. If VQ ratio in this some diseased area of the lung, VQ ratio is 10, right? There's a diseased area. Yes, VQ ratio is 10. So this will result in an increase in physiologic dead space. Why? Because this is an area where there is a wasted ventilation, right? Then VQ ratio more than 1 is a wasted ventilation. Okay? So this will become increased dead space. Okay? Now, please remember, normal individuals, wasted ventilation is 0. In a normal individual, wasted ventilation is equal to 0. There is no wasted ventilation. Yes? So normally, the physiologic dead space is equal to the anatomic dead space. Normally, there is no wasted ventilation, right? Normally, there is no wasted ventilation, so physiologic dead space is equal to the anatomic dead space. If there is a wasted ventilation, either because of overventilation in certain areas, or it could be because of underperfusion in certain areas. Underperfusion kaise ho sakta hai? If there is a thrombus which is blocking a blood vessel, maybe a partial block, underperfusion, right? Then you will get a wasted ventilation. Normally, there is no wasted ventilation. So wasted ventilation is 0. So physiologic dead space is equal to the anatomic dead space. Next important point. How do you measure the anatomic dead space? How do you measure the physiologic dead space? Anatomic dead space is measured by, the Fowler's technique. This is measured by the Fowler's technique or the Fowler's method. Again, this has been asked as a question. Fowler's technique or Fowler's method. And what is this Fowler's technique or Fowler's analysis in the expired air? Nitrogen analysis in the expired air after a single deep breath, of 100% oxygen, after a single deep breath of 100% oxygen, right? Measured by the Fowler's technique. What is this? Nitrogen analysis in the expired air, single deep breath of 100% oxygen. This is also known as single breath nitrogen analysis. Single breath nitrogen analysis. Right? This is nitrogen analysis of the expired air after a single deep breath of 100% oxygen, which is also known as the single breath nitrogen analysis. Okay? This is your Fowler's technique for the measurement of anatomy. What is anatomic? See, dead space is anything, air which is not participating in respiratory exchange, right? This is the air which is present in the trachea, bronchi. There is no exchange taking place in the trachea or the bronchi, at least not in the larger bronchi. Terminal bronchioles, yes, but not in the, what is known as the conducting zone of the lung, isn't it? There is no exchange taking place. So that becomes your anatomic dead space. What is physiologic dead space? Physiologic dead space is anatomic plus wasted ventilation. I've discussed with you what is wasted ventilation, right? Wasted ventilation is when the VQ ratio is more than 1. That is called wasted ventilation. Now, what is physiologic dead space? Now, how to measure physiological dead space? Physiologic dead space is measured by what is known as the Bohr's equation. This is met by the Bohr's equation. And for Bohr's equation, I need three parameters. I need three parameters. Bohr's equation. Bohr's equation is my nitrogen analysis when we are taking 100% oxygen. Single deep breath of 100% oxygen. Normally you're breathing room air, which has got oxygen, nitrogen. Then you take one deep breath of 100% oxygen. What will come out? Nitrogen from the previous breath. Okay. समझ आ गया मधुरा, भावित, Vivek? Bohr's equation. Now, as far as Bohr's equation is, for Bohr's equation, I need three things. Three things is partial pressure of CO2 in the expired air. E is expired air, right? Partial pressure of CO2 in the expired. I need partial pressure of CO2 in the arterial. Partial pressure of CO2 in the artery. P stands for partial pressure. Capital E is expired air, arterial blood, right? Small a is arterial blood. And I need VT. What is VT? This is tidal volume. If I have these three measurements with me, I can use the Bohr's equation. These three. And this was again asked as a question, and that is the if I know these three parameters, what can I measure? I can measure the physiologic dead space. I need partial pressure of CO2 in the expired air, partial pressure of CO2 in the arterial blood, and the tidal volume. Right? So this is called this is Bohr's. What is the Bohr's equation? Bohr's equation says, now VD is the physiologic dead space, right? Dead space. Now VD by VT ratio is equal to partial pressure of CO2 in the arterial blood minus the partial pressure of CO2 in the expired air divided by partial pressure of CO2 in the arterial blood. This is your ratio of dead space by tidal volume. What is this? PACO minus PECO divided by PACO. PACO minus PECO divided by PACO. This is Bohr's equation. This gives you the ratio between dead space and tidal volume. PACO minus PECO divided by PACO. This is, and how much is the normal dead space by tidal volume ratio? This is 0.33 to 0.40, 33 to 40%. That means the dead space should be approximately one third of the tidal volume. One third of the tidal volume. Okay? Again, this has been asked as a question. Dead space is one third, one fourth, one fifth. So this is one third. Yes, PACO, PECO. Okay, PECO. All right. This is, again, let's see, there is a physiologic and the anatomic dead space. How do you measure anatomic dead space? This is by the Fowler's technique. What is Fowler's technique? Nitrogen analysis in the expired air after a single deep breath of 100% oxygen, which is also known as the single breath nitrogen analysis. Yes? Bohr's equation is used to measure the physiologic dead space, but for these you need three things, and that is PeCO2, PaCO2, and tidal volume. What is the Bohr's equation? Dead space by tidal volume ratio is PACO minus PECO divided by PACO. And how much is the normal dead space by tidal volume ratio? 0.33 to 0.40, or one third of the tidal volume. Okay? So this is as far as your physiologic and the anatomic dead space is concerned. Now, we've already discussed that if the VQ ratio is more than 1, if the VQ ratio is more than 1, yes, VD is dead space, yes. If VQ ratio is more than 1, we said this is a wasted ventilation. Yes, I've already discussed it with you. Wasted ventilation. If there is a wasted ventilation, that means there is now an increase in the physiologic dead space. I've discussed it with you that whenever there is a wasted ventilation, that means increase in the physiologic dead space. Now let us understand, what do you understand by the VQ ratio less than 1? VQ ratio is less than 1. VQ ratio is less than 1. What does it mean? It means that the ventilation is less than the perfusion. Numerator is less than the denominator, right? So that means now, this means this is a wasted perfusion. Now, perfusion is in excess of the ventilation. Have you understood? Perfusion is in excess of the ventilation. Is in excess, or ventilation is less than the perfusion. In other words, it is a wasted perfusion. That means the ventilation is now inadequate or not enough to oxygenate the blood. So this blood flow is being wasted. It is a wasted perfusion. And whenever there is a wasted perfusion, that means there is going to be an incomplete oxygenation of the blood. There is an incomplete oxygenation of the blood, and whenever there is an incomplete oxygenation of the blood, this is known as shunting. This is known as shunting. Please understand, a VQ ratio of 1 is an ideal VQ ratio, maximum oxygenation. VQ ratio more than 1, ventilation more than the perfusion, it is a wasted ventilation. Increases the physiologic dead space, right? VQ ratio less than 1 means now the perfusion is less, ventilation is less, perfusion is more. It is a wasted perfusion. This is an incomplete oxygenation of blood, which is known as shunting, right? So this is as far as your VQ ratio more than 1 and less than 1 is concerned. We've already discussed, so please remember, wherever the VQ ratio is less than 1, this is incomplete oxygenation, and whenever the VQ ratio is more than 1, that means increase in the physiologic dead space. Okay? All right, so this is your anatomic dead space by Fowler's technique, measurement of physiologic dead space by the Bohr's equation. Bohr's equation. Next, let's go on to the what is spirometry? Yes, physiologic dead space, 1/3 of tidal volume. Yes. Okay. All right. Bit of ventilation, VQ ratio less than 1. Let's see. Let me just try and tell you this here. VQ ratio more than 1, like I said, pulmonary embolism. VQ ratio will increase. Yes. Ventilation less than 1, ventilation of 0, VQ ratio, pulmonary embolism, VQ ratio is in fact infinity. Perfusion is 0, right? Pulmonary embolism. Foreign body. If there is a foreign body in the lungs, what will happen to the VQ ratio? VQ ratio will become 0. No ventilation in that particular area. Foreign bodies lodged in, let's say, a major bronchus. The ventilation reduces, becomes 0. So VQ ratio will become 0. Have you understood, right? So this is the, okay. All right. Next, let's go on to what is spirometry. Let's discuss spirometry. Please stop the unnecessary questions. It is distracting. I lose out on the genuine queries, and there are going to be genuine queries, right? So unnecessary, chhod do beta. Mimamsa ke piche mat pado, chhod do. There is nothing to Okay, let's just concentrate on the work and just put in your genuine queries and don't cross talk with each other. It sort of distracts me and then it's going to affect our studies. Okay, chalo, let's see spirometry. Now, what happens in spirometry? Now, what is spirometry? This is lung volumes and capacities. Lung volumes and capacities. Now, lung volumes and capacities, let's try and see this. Right, lung volumes and capacities. The first one is what is known as the tidal volume. Tidal volume. Let's see what is tidal volume. Tidal volume, this is the volume of air. Volume of air inspired or expired. Volume of air inspired or expired during a normal, quiet respiration. This is the volume of air inspired or expired during a normal, quiet respiration. Right? बेटा अनिल मैंने बोला तो है Fowler's technique is for anatomic and physiologic includes wasted ventilation. उसके लिए Bohr's equation है. क्यों confusion हो रहा है? मैं लिखा तो है और 10 बार repeat भी किया है ना? The anatomic dead space, Fowler's technique. Physiologic dead space is Bohr's equation. तो क्यों confuse हो रहे हो बेटा? Tidal volume, volume of air inspired or expired during a normal quiet respiration. This is known as tidal volume. And tidal volume is the only volume which is the same in males and females. This is 500 ml in males and 500 ml in females. यशवंत क्या लिख रहे हो बेटा? VQ ratio is 1/3 of tidal volume. VQ ratio is ventilation perfusion ratio, है ना? Ventilation मतलब the amount of air going into the lungs. Perfusion मतलब the blood flow in the pulmonary blood vessels, isn't it? That is VQ ratio. Now, we said dead space. Dead space is 1/3 of the tidal volume. What is dead space? Dead space is the volume of air which is not participating in respiratory exchange. ठीक है? जो डेड स्पेस है दैट इज 1/3. टाइडल वॉल्यूम कितना होता है? 500 ml. डेड स्पेस कितना है? 140 to 150 ml. So it is approximately 1/3 of the tidal volume. 500 ml of air जो हम ब्रीथ कर रहे हैं उसका 1/3 हिस्सा does not participate in respiratory exchange. That is what is meant by dead space by tidal volume ratio. ठीक है? All right. Please, nobody is going to be nasty here. ठीक है? We are this is something which which is something which I don't approve of at all. All of us can have genuine doubts, isn't it? We can have, and there is so much information which is going in one day. There is bound to be confusion. Yes, I will choose which questions to answer, but let's not put each other down. We have to help each other and try and achieve our ultimate goal. But don't put each other down. No, I don't like it. That's not nice. All right. So what is tidal volume? Volume of air inspired or expired during a normal quiet respiration. Now, next is what is inspiratory reserve volume? What is inspiratory reserve volume? This is IRV. Inspiratory reserve volume. Yes. What is inspiratory reserve volume? Let's try and see this. Inspiratory reserve volume, this is the volume of air. Volume of air inspired over and above a tidal inspiration with maximum effort. Over and above a tidal inspiration with maximum effort. This is called IRV, inspiratory reserve volume. Inspiratory reserve volume. This is over and above a tidal inspiration with maximum effort, that is known as IRV. Over and above a tidal inspiration with maximum effort. This is what is known as IRV, inspiratory reserve volume. This, how much is inspiratory reserve volume? Inspiratory reserve volume in males is 3300 ml, in females about 1900 ml. There is a difference in the IRV in males and females. Males have larger thoracic cages, more muscle mass, so they are able to inspire more over and above a tidal inspiration. Females, the IRV is 1900 ml. Yes. This is your tidal volume. And what is tidal volume? Volume of air inspired or expired during a normal tidal inspiration, right? And IRV over and above a tidal inspiration with maximum effort. Then next, let's go on to what is ERV. What is ERV? Expiratory reserve volume. What is expiratory reserve volume? Volume of air. Volume of air expired forcefully. Volume of air expired forcefully over and above. Volume of air expired forcefully over and above a tidal expiration. Over and above a tidal expiration with maximum effort. Over and above a tidal expiration with maximum effort. This is known as ERV, volume of air expired forcefully over and above a tidal expiration with maximum effort. Over and above a tidal expiration with maximum effort. So if this is the tidal volume, yes, this is the tidal volume, I ask him to breathe out forcefully. Volume of air which is able to expire forcefully over and above is known as ERV, right? Over and above is, you ask him to forcefully expire, yes? So ERV in the case of males, ERV in the case of males, this is about 1000 ml. In the case of females, about 700 ml. Right? In case of males, 1000 ml, females, 700 ml. Then we go on to known as residual volume. Residual volume. And what is residual volume? This is the volume of air. What is residual volume? This is the volume of air which remains in the lungs. Volume of air which remains in the lungs. At the end, maximum expiration. Volume of air which remains in the lungs at the end of a maximum expiration. Volume of air which remains in the lungs at, even when I do a forceful expiration, my lung volume is not zero. Volume of air which remains in the lungs at the end of a forceful or a maximum expiration, that is known as the residual volume. So that means if you look at this over here, this becomes the residual volume, right? Volume of air which remains in the lungs at the end of a maximum expiration, that is known as the residual volume. And how much is the residual volume? This is 1200 ml in males and 1100 ml in females. 1200 ml in males and 1100 ml in females. Okay. Now let's have a look at the capacities, right? Let's have a look at the capacities. The first one here is, what do you understand by inspiratory capacity, right? What is inspiratory capacity? What is inspiratory capacity? Inspiratory capacity is tidal volume plus IRV. That is the inspiratory capacity. Right? Inspiratory capacity. राजेश शर्मा बेटा definitions के ऊपर ध्यान दो. Residual volume क्या है? volume of air which remains in the lungs after a forceful expiration. Supposing I do a forceful expiration. At the end of forceful expiration is residual volume. What is the anatomical dead space? Whatever air that we are breathing, right? When we breathe in air, there is some air which will be present in the airways. This cannot take part in respiratory exchange. Exchange kahan hota hai? Alveoli par. Exchange with the blood. There is no exchange of gases in the from the air in the trachea, major bronchi, right? So that is known as dead space. So दोनों की definitions पर ध्यान दो. हमेशा have a look at the definitions. Remember the definitions. Once you know the definitions, then it becomes clear, right? Residual volume is the volume of air remaining in the lungs at the end of a forceful expiration. If I told you forcefully expire karo, lung volume आपकी zero नहीं रहेगी. There is, in case you're a male, your lung volume will be 1200. That is the residual volume. Dead space is the air which is not participating in respiratory exchange. Okay? So then is expiratory capacity. Expiratory capacity. What is expiratory capacity? Expiratory capacity is tidal volume plus ERV. Inspiratory capacity is tidal volume plus IRV. Expiratory capacity is tidal volume plus ERV. So if you look at this, this becomes the expiratory capacity. Tidal volume plus the ERV. This is expiratory capacity. Then comes, what do you understand by functional residual capacity? Right? Functional residual capacity, FRC. FRC, this is known as the functional residual capacity. Functional residual capacity, FRC. What is FRC? This is the volume of air which remains in the lungs. Volume of air which remains in the lungs at the end of if a normal or tidal expiration. At the end of a normal or a tidal expiration, right? FRC, volume of air which remains in the lungs at the end of a tidal expiration. This is known as FRC, functional residual capacity. Volume of air which remains in the lungs at the end of a normal or a tidal expiration. Yes, that means if you look at this over here, this is your tidal volume, right? Volume of air which remains in the lungs at the end of a normal or a tidal expiration. This is called FRC, functional residual capacity. Right? That means at the end of a normal expiration, the volume of air which is present in your lungs will be equal to 2200 ml in the case of a male, and about 1800 ml in the case of a female. That is called functional residual capacity at the end of a normal or a tidal expiration. Okay? Then you come to what you understand by forced vital capacity. What is vital capacity or forced vital capacity? Or forced vital capacity, right? Vital capacity or forced vital capacity. This is the volume of air expired forcefully. Volume of air expired forcefully after a forceful volume of air expired forcefully after a forceful inspiration. That is vital capacity or forced vital capacity. What is functional residual capacity? This is going to be ERV plus the residual volume. What is vital capacity? Vital capacity is IRV plus tidal volume plus ERV. Right? This is the volume of air expired forcefully after a forceful inspiration. Right? So that means how do you see the vital capacity? It's very easy. Vital capacity, you ask the patient to take a maximum inspiration. Maximum inspiration, right? You ask the patient to take a maximum inspiration, right? And now you ask him to breathe out forcefully. And when he breathes out forcefully, he will first breathe out his IRV. Yes. Then he will breathe out his tidal volume. Then he will breathe out his ERV. Yes. This is your complete vital capacity. What is left inside the lungs will be residual volume. He will breathe out his IRV plus tidal volume plus ERV, and together this is known as the vital capacity or the forced vital capacity. This is known as the vital capacity or the forced vital capacity, right? And what remains the lungs at the end of a forced expiration is known as the residual volume, right? So then one more capacity is left, and that is the total lung capacity. Add all the lung volumes, you will get total lung capacity. Total lung capacity in the case of males is 6,000 ml. In the case of females, 4,200 ml. Add up all the lung volumes is going to be total lung capacity, right? So this is as far as your lung volumes and capacities is concerned. First, have a look at tidal volume. What is tidal volume? Volume of air inspired or expired during a normal quiet expiration. Sorry, quiet respiration. IRV, volume of air expired over and above a tidal inspiration with maximum effort. Volume ERV, volume of air expired forcefully over and above a tidal expiration with maximum effort. Residual volume, volume of air which remains in the lungs at the end of a maximum expiration. Inspiratory capacity, tidal volume plus IRV. Expiratory capacity, tidal volume plus ERV. Functional residual capacity, ERV plus RV. Vital capacity, IRV plus tidal volume plus ERV. At the end of vital capacity, the volume of air which remains in the lungs is known as the residual volume, right? Now, one more important point, and that is, if you see this here, if this is zero lung volume, this is the lung volume at the end of a maximum expiration, which is going to be residual volume. And residual volume in the case of males is 1200 ml, right? This is residual volume here. This is 1200 ml. And 1100 ml in the case of females. And what is this residual volume? Residual volume is the lung volume at the end of a maximum expiration. Lung volume at the end of a maximum expiration, this is the residual volume. What is the lung volume at this point at the end of a normal expiration? This is functional residual capacity. And how much is functional residual capacity? This is 2200 ml in the case of males and 1800 ml in the case of females. What is this functional residual capacity? This is the lung volume. Lung volume at the end of a normal expiration. This is the lung volume at the end of a normal expiration. This is known as functional residual capacity. Have you understood? Lung volume at the end of a maximum expiration, residual volume. Lung volume at the end of a normal expiration, functional residual capacity, right? When you take tidal inspiration from FRC, that means you add 500 ml of air. So now the lung volume becomes 2700 ml in the case of males. You're adding 500 to 2200, and in females 1800 plus 500 will become 2300 ml, right? Now I ask you to take in maximum inspiration. Inspire forcefully. Males, that means inspire your IRV. So at this point, when you forcefully maximum inspiration, this is going to be your total lung capacity. This is 6000 ml because you've added your IRV, 3300 ml in the case of males and 1900 ml in the case of females. So 4,200 ml in the case of females. What is this total lung capacity? This is the lung volume. Lung volume at the end of a maximum inspiration. Forcefully inspire. You're forcefully inspiring, you're increasing the volume of the lungs, and at the end of a forceful expiration, your lung volume will be total lung capacity. From a maximum inspiration, if I ask you to breathe out forcefully, you will breathe out your vital capacity. The only way to remember these is remember the definitions, right? Again, all these three questions have been asked in different NEET exams in the last 3 years. Lung volume at the end of maximum expiration, residual volume. Lung volume at the end of a normal expiration, FRC. Lung volume at the end of a maximum inspiration is the total lung capacity. Volume of air expired forcefully after after a forceful inspiration. Volume of air expired forcefully after a forceful inspiration. This will be equal to the forced vital capacity. This will be equal to the forced vital capacity. Yes, this is your. These are the 4 important questions which have been asked in NEET exams. Chalo, so lung volumes and capacities. Okay, go through this. You can take a break. Are we due for a break? Yeah. Okay, you can take a break for 10 minutes till 6:15 and then we can continue. Okay. Abhishek Patil, residual volume तो करते ही नहीं है ना, expire कर ही नहीं सकते. Lung volume cannot be 0. Residual volume is the minimum volume of air which remains in the lungs. Lungs के अंदर जो बच जाता है, that is called residual volume. ठीक है? Okay. Okay. हाँ जी बेटा 10 मिनट का ब्रेक दे दो. Okay. क्या चीज? 6:15 पे. हाँ ठीक है. ओके.